Today I'm going to be talking about describing you basically the progress we made over the last 2.5 years within the first Russian ECoG-based BCI project. This project is supported by the mega grant that has been won in 2018 by Mikhail Lebedev. And in response to this grant, High School of Economics has created a Center for Bioelectric Interfaces that is situated now in the center of Moscow. And we are doing this work, and in the parallel, we are doing a lot of accompanying projects, part of which I will tell you about now. Okay. So the basic idea of the project is to create a bidirectional ECoG-based neurointerface, and we had 3 years, and we basically had 3 tasks. The first task would be to create a decoder from the motor cortex, then to learn how to stimulate sensory cortex in order to provide some sort of sensory feedback to the subject. And then in the 3rd year, we were supposed to merge the first and the second goals and to create the bidirectional neurointerface. Okay. So let's see so now we are many. We have collaborators from different places in St. Petersburg and from clinical labs in Moscow, and we're also collaborating with some additional departments from HSC. So total we have now about 22 affiliates, out of them 7 hold PhDs, and we have 2 Doctor of Sciences, and our team is pretty young. Many people are younger than 35. Okay, so we established 2 clinical partners because this project cannot be done without them, and because it's very interesting and fruitful to interact with the clinic. So the first one is the Polenova Institute in St. Petersburg, and Mikhail Alexandrov and Nastasia Arkhipova, the doctors there. And the second one, and I would say the main base that we are actively working with now, is that in the 3rd Medical University in Moscow, and its university clinic led by Vladimir Krylov, and the newly established lab for invasive neurointerfaces that HSC and the 3rd Medical University fund together, and it's led by Mikhail Sinkin, okay, a neurologist. Okay, so the first goal is kinematics decoding. So it's pretty simple in terms of conceptual setting. So we have ECoG recordings recorded from the ECoG grid implanted over the motor cortex, and we have certain kinematic parameters, let's say finger velocity, and we need to decode it. Okay, so in order to do that, in order to do research in this field, we set up an experimental setup, which comprises the microgrids, comprises the software, which merges together the data from EEG, from ECoG amplifier, from motion capture system, and basically in video stream, and then collect the data, and if needed, streams them to the high performance servers for decoding and for other online or offline analysis. Okay, so we arrange the work so that before we interact with the patient, we first streamline everything using the EMG, electromyographic recordings. On the one hand, it's easy and interesting. It's interesting and very, very fruitful to actually prepare for the visit to the patient. And on the other hand, the EMG interfaces in itself a very, very promising field, and maybe in let's say 80% of patients, even 90% of patients, you don't really need to go into the brain and simulate the brain or decode from the brain. You can stimulate peripheral nerves, and you can record from muscles, okay, in order to activate the prosthetics. So here is how our decoder works. See, the movements are smooth, and you don't have to do any extra force to do the movement. On the right hand side is the real hand, the motion capture signal, and on the left hand is decoded. We have also some videos on our website where you can play piano with this kind of interface, and we record the data with 64 channel EMG grid, and it's basically 4.5 centimeters squared area, And we can you can say that, okay, maybe it's not really needed, so many channels are not really needed, but in fact we can obtain up to 10 independent components from this small area, which I think is a very interesting and promising approach to read off muscle activity non-invasively. But anyway, this whole thing within this project is done primarily to prepare for the visit to the patient. And we developed decoders, which I'll show you how to perform, but also we focused on not only the decoding itself, but also on developing the decoders which would allow us to interpret the parameters of decoding. Okay, so in order to understand which neuronal populations are harnessed in order to provide the decoding and the kinematic parameter, which neuronal population code these, we started developing these decoders from basic principles, which are the following. Okay, so when we have a grid of electrodes superimposed on the cortical surface, it records populations of neurons, the local field potentials produced by the activity of these populations, and from it's a well-known fact that the activity of these populations maps onto the sensors via a linear model. So in order to get access to a particular population, then we need to apply also a linear unmixing that would extract the activity of this particular source, right? This is one story. This is one known piece. And the other known piece that has already been mentioned, that the motor activity is accompanied by the changes in the rhythmic activation patterns of the brain. So in particular, higher frequencies like gamma tend to synchronize, and low frequencies like alpha and beta tend to desynchronize. And what's interesting is that the alpha desynchronization, being the lowest frequency out of these three, tends to occupy a larger area as compared to beta, where beta is a higher frequency and the area is smaller, and the gamma synchronization happens in a very, very tiny spot, which allows us to more specifically localize, for instance, motor cortex related to the finger, find specific finger representation. Okay, so the phenomenological model of the design is the following. So we have a set of populations. These populations somehow act and influence the activity of muscles, which move the hand, the wrist, the fingers. And this very activity of these populations is recorded by the electrodes. The problem is that the electrodes record not only the populations which are important for the finger movement and hand movement for the specific motor task we are interested in, but it also records the activity of other populations, okay? And therefore, the measured data can be represented as a sum of the target activity and some non-target interference activity, which nevertheless leaks into the sensors, of course. So our goal is to find such mapping from X into the kinematics, and also interpret F in order to find the topographies, which would allow us then to localize the sources, and to find the spectral characteristics of temporal activations of these populations that would allow us to fit some sophisticated dynamical models and to understand the functioning of the entire process. Okay, so we attempted it by just creating this compact architecture that basically in the first layer does unmixing, which is a simple linear unmixing. Then we have a bandpass filter. Then we have a bandpass filter, and then we have the extractor of the envelope, and then the samples of the envelope are mixed together with the fully connected layer. Okay, the reason why we selected this kind of architecture, well, first of all, because each branch of this architecture is doing exactly something to counteract the mixing effect, which I just mentioned in the beginning, and also it's isolating the rhythmic activity and then making decisions based on this rhythmic activity, okay, based on the intensity of this rhythmic activity. That means that this kind of architecture is potentially interpretable. So we can, for instance, look at these spatial filtering weights and understand where the signal is coming from. We can look at the bandpass filter properties, or in general frequency domain filter properties, in order to get insights into the dynamical properties of activity of the population. Okay, so in general, if you think about these first two layers, the processing is spatial filtering first, the spatial filter multiplied by your data, and then convolved with some temporal filter. Here it's applied to a chunk of data, therefore convolution is just replaced by a product. Okay, so this architecture works pretty well. We applied it to our data, and we also applied it to the Berlin BCI competition. And as you can see, that our solution performs on par, maybe slightly better, somewhere slightly worse, but it does not, as compared to the winner of this competition, and it does not require any feature engineering. Okay. On the contrary, as I will show later, it allows interpretation of the weights and allows to get insights into the dynamic and spatial properties of the pivotal populations. Okay, so I can show you this example how it works. So this is a patient. She was given this pen just to help her. It doesn't click, but when you see this is the finger she's moving, and the decoding result is here. Now she's doing it without the. So the pace is repeated, and so yeah. So in order to make this whole thing work, however, we had to do iterative learning, meaning that basically what happened is this: when we first recorded the data from this patient and trained the model, and then we applied this model to the in real time to the new chunk of data, it just simply didn't work. And we realized that we had to train the patient to make the patient do the movements consciously. So we were showing to the patient certain profiles she had to follow with her finger movements, and gradually her performance improved. So we recorded one such chunk of data when she was following the certain profile. Then we created a decoder. Then we repeated this same process 3 or 4 times, and then we arrived to the decoding accuracy of about 80%. And you can see that this decoding is without a lag. Okay, which is interesting and I think very promising. Okay, so here the black one is the true movements, and the blue one is the decoded trace. Okay, but the main goal is not even to create this decoder, but also to create an interpretable, to create an interpretation of the weights the decoder learned. Okay, so I will not go into the mathematical details here, but I'll just try to give you the intuition behind this whole thing. When in this architecture, the temporal filters, those providing frequency domain selectivity, and the spatial filters are learned, they are both not only trying to align themselves or align with the target source, but also they're trying to tune away or orthogonalize themselves from the interference. So when interpreting the weights, you have to take this into account, and this problem has been addressed in 2014 in a famous paper by Stefan Halfe when he applied it to a simple regression problem. Here we have a different problem. First of all, it's not a simple regression. We are, you see, we multiplying our data on both sides, so it's sort of a quadratic thing. And also we have multiple branches. So when interpreting spatial weights, we have to take into account the temporal context, the frequency domain context, which is provided by the temporal filter. Okay, and on the contrary, when interpreting temporal filter weights and looking at seeking power spectral densities of activity of our populations, we have to take into account the specific spatial properties, spatial filter. Okay, so we played with all this. We done simulations which show that the developed approach works properly. You see the blue and yellow lines coincide. This is a true pattern that was simulated, and the recovered pattern with yellow. Okay, so, and other approaches do not work as accurately. And also then we did Monte-Carlo simulations, and we showed that the blue ones, the spatial weights are recovered very nicely, and temporal weights also recovered much better than the competing solution. Okay, so we believe that this approach can be used to to uncover the spatial and dynamic properties of the populations pivotal to decoding, which can be applied for instance, eloquent cortex mapping when the patient is involved in a motor task and we are looking for the brain areas for the cortical regions representing specific fingers, for instance, like it's shown here. So we can see the locations, the topographies recovered. We can see that they agree with somatotopy. So the pinky is above, and the thumb occupies larger area and largely below. And also what's interesting is the temporal profiles. See, while the filters do emphasize higher frequencies, okay, if you look at the pattern of the activity, the spectral pattern of the activity of the pivotal population, you oftentimes get low frequency components like here, and the fact that filter emphasize higher frequency doesn't mean that that there is no information in the low frequencies. It just means that there are other populations which are active in these low frequencies, and the filter is just trying to tune itself away from the activity in the low frequency because it's mixed together with non-informative components, and therefore it basically tunes itself away from them. Okay, so this is, and then these models, these blue curves, for instance, can be fitted with some physiological models, which we are now in the process of playing with, together with the approach which was developed by Stephanie Jones in her simulator. Okay, so, and also this has a direct application to the intraoperative passive speech mapping, which we are also developing with our clinical partners. So the idea here is that we record the activity during picture naming task, and then analyze, or either look for these patterns or fit simpler models to determine which activity of which electrodes deviates during the task as compared to the rest period. Okay, and so far we have very good agreement, both in true positive and true negatives, and we are collecting more subjects, more data with our clinical partners now. Okay, so you can see about the poster about this work more, and you can talk about Artur Petrosyan, our PhD student. He can talk more about it. Now I will go to the second task. I will speed up a little bit here. So the second task was about sensory component of the interface in order to substitute the afferent pathway. So we developed a active touch paradigm in which subject has to explore the textures, but he or she didn't see the textures. So this here, the video, it just shows you the texture, but in fact the subject didn't see them. And every time the subject crossed the line, he or she would receive a electrical stimulation of the cortex or of the finger. So we implemented both peripheral stimulation and direct cortical stimulation. Okay, so in order to do that, we first tried to find the cortical region in which we will be able to elicit sensory percept in an index finger, and in this process we discovered one interesting thing, that we played with patients who were implanted with microgrids and with macrogrids, and we found that the patients with microgrids were more likely to, as a result of stimulation, to produce sensory percepts, the orange bars, as compared to the microgrids, where the dominating reaction was motor reaction. Okay, this is interesting. This shows a potential promise of microgrids for eliciting sensory percepts, and also with proper analysis and proper modeling of the brain tissue and physiology of the multi-layered structure may give additional insights into organization of the sensory circuits, okay, in layer-wise organization. Okay, so then we had a patient, so far only a single patient, and unfortunately this patient was to be implanted with only a stripe of electrodes, and it was not a microgrid, and the distance between electrodes was 1 centimeter. So we first did MEG, and we stimulated the index finger, and we localized with and analyzed the magnetoencephalographic data and localized the sensory cortex, which is where the index finger is represented. Then we did MEG informed implantation, the doctors did this implantation. So we were pretty accurate because stimulation of the between the third and the fourth electrodes, as we that determined, was causing sensory percepts in the index finger. Okay, and then we compared with this patient we did peripheral stimulation. So basically just directly the finger when he was moving, when he was exploring the texture. And we did cortical stimulation. So we had a technical problem with this third texture. We discovered it, you know, already after we done it. So with cortical stimulation, we get about 67% of accuracy in this binary classification task where the subject has to decide which texture is denser, okay? And in peripheral stimulation, we got about 80% of accuracy. So the reason why this accuracy is not that high can be due to the fact that the grid is not microgrid. And also we encountered habituation in a way that as the session progressed, we had to increase the current, the stimulating current a little bit, step by step. And after crossing some threshold, the subject started to feel started to generate motor reactions in addition to sensory percepts. So that all kind of confused the subject, and therefore, you know, this is the first experiment of this of ours with this cortical stimulation, and we're hoping to get another subject to continue that work. Okay, you can see the poster by Maria Volodina, our research scientist, which describes exactly this in the poster session. And also, well, the third task is to implement the bidirectional ECOG interface. Here we implemented a virtual reality scenario. We developed our own stimulator that can be controlled through the computer, and it can deliver not only stimulation between two selected pins, but it can also deliver distributed in time and space stimulation at a fast pace. Okay, and now we have streamlined it with EMG, but we are waiting for the patient, and because of the COVID, we haven't had a patient for half a year. So here is the scenario. I just show you the scenario. For instance, a successful attempt would be to collect this fragile Christmas balls. So basically the subject controls the grasp, and then if when he or she touches the ball, he gets feedback, only sensory feedback, because as you can see, the index finger is not seen, it's hidden. Then he has to stop pressing and do the action. At the same time, when you are pressing too much, like here, the ball drops. Okay, so this is the task that our subject has to fulfill, and we hope to be able to do it by the end of the year, but it all depends on the availability of the patient. Okay, so we are also developing, as I already mentioned, immediate clinical applications. One of them, as already mentioned, the speech mapping, passive speech mapping. The other one is lower limb controlled exoskeleton for the neuro rehab purposes. Here is the exo-athlete. He is a paraplegic. He's leaning against crutches, generating severe muscle artifacts, which we are coping with, which our interface is coping with. And the interface is basically, the subject is asked either to stop, either to stay still, or to do the movement, because we wanted to test both the specificity and sensitivity. And it seems to work pretty nicely. We get about 70–75% of ROC, area under ROC curve in patients. We had already 3 patients, 3 true paraplegic patients we played with. And we are developing this technology with Exo-Athlete company, and we hope now to get some additional funds to proceed with this topic. Okay. So also I would like to draw your attention to other research that is going in our center. We are improving the motor BCI using the EEG inverse problem solution, and we get very promising results. We're also active in the field of analysis of MEG, magnetoencephalographic data, for decoding the coupling, for trying to localize coupled sources in the brain and develop statistical tools for that. We have also done some significant work on neurofeedback, and we are exploring what low latency neurofeedback gives us. So far, we see that it improves the learning, it speeds it up, and makes the sustained effect stronger. then also Masha Volodina had this very nice study where we really saw that EEG is informative about the way people meditate, and especially those which are experienced ones. We can see that there's delineation between two different groups of experienced meditators, but she will have a talk, so you can listen to the talk. Also, there is more fundamental work on perception of time when you're listening to the musical pieces, and familiar and unfamiliar musical pieces. And also this novel work with order-based brain-computer interface developed by Ivan Ninenko together with Mikhail Lebedev. Another work that is maybe not presented here, but it will be shown in other posters that you can see in the conference. So, so I'd like to conclude now that the working on the task of building decoders of ECOG into motor activity, ECG data into motor activity, we can not only do this decoding, but we can also, with proper interpretation of the weights of the proper architectures, we can actually extract knowledge about the neurophysiology of the motor control. Okay, and this can be done with compact convolutional neural networks. That one of the solutions I've just showed to you. Okay, we applied this technology to motor speech mapping and motor cortex mapping, to passive speech mapping and motor cortex mapping. When you're doing the stimulation via microgrids, we found that microgrids are more likely to generate sensory percepts, which is a potential insight. And also recent works, not by us, but by other groups, show that these microgrids can actually be used to navigate the places where to implant the probes for the stimulation and for decoding in patients, in paraplegic, quadriplegic patients. Okay, when implemented cortical active touch, we obtained about 67% of texture classification accuracy. The problem could be the low accuracy, this relatively low accuracy could be due to the use of macrogrid that happened due to medical reasons in that particular patient. And what I'm particularly excited is that while it's quite clear that ECOG interfaces themselves cannot be used for the real control of the prosthetic devices, just because, you know, these ECOG grids are not designed for that. Okay, so but the ECOG grids record activity not of single neurons, but activity of large populations of neurons and local field potentials, basically. And when such electrodes, biocompatible electrodes having the same properties, recording the same type of data, are developed, the technology that we develop in analyzing the data can be applied to then to transfer these technologies into real motor control and by myoelectric control, you know, the prosthetic control. So, and as I mentioned, we're also developing non-invasive BCI solutions for near rehab purposes. Okay, so basically that's it.